So let's start. Welcome everyone to this very first Triple Helix Expertise Exchange workshop on the modeling of drug-device interaction. The reason we started to organize this workshop was because there is a flurry of activity in the domains of regulatory science for devices and drugs, and especially in the area where they connect. It's probably time to also look at how to do that validation of those models. The reason we chose this kind of triple helix format is because this really is to open the discussion and to really start the communication and define or identify the interested partners, but also the questions that need to be answered. So what we have done today is brought together three groups of speakers and participants, being academia, regulators, and industry. And for those that know the Avicenna Alliance and the VPH Institute, we've used the orange for academia, the Avicenna blue for industry, and the dark blue for the regulators, just so that you can immediately place where the different speakers are coming from. What we have is a program where in a minute I'll stop talking and then I'll give the floor to 4 technical experts, 2 from academia, 2 from industry, also 2 from Europe and 2 from the US. And after that, we'll give the floor to 2 regulators. Both are national regulators in Europe and member of the Modeling and Simulation Working Party of EMA. After that, there should be enough time for a discussion and to answer questions. Questions that you are very welcome to post in the chat during the presentations. After every presentation, there is a few minutes to answer specific questions on the topic, but the broader questions and discussion items, we would like to keep them for the end. Every time that I introduce a new speaker, there will be a small poll that will pop up. That is just our way of trying to probe the audience that we have here today, where you're from, what kind of expertise you have, or what role you have in your organization, so that we can communicate that at the end of the workshop, and that we can see if there maybe are interesting things to conclude from that. You've also received a link to a survey that we are going to use to already derive some questions from for the panel discussion, but that we will circulate again in the email thanking you for your participation that will come after today. And that will be also a way to identify people that are interested in follow-up activities and engaging in this particular topic and collaborating with the regulatory agencies. So that is it for what I had to say. I will now introduce, maybe Federica, you can share the first poll. And it's my pleasure to introduce as first speaker, Yu Feng from the School of Chemical Engineering of Oklahoma State University. He's also an investigator of the Oklahoma Center of Respiratory and Infectious Diseases. He had his degree from North Carolina State, where he also worked as a professor before going to Oklahoma. And his group is really focused on making contributions in the area of patient-specific pulmonary health problems through the use of multi-scale modeling techniques. So thank you very much for coming. The screen is yours.
Yeah, thank you, Liz, for this opportunity to let me share our research experience. So yeah, let me share my screen first. Yeah, I would like to take this opportunity to share our research experience with building the what we call all-in-one modeling framework to predict long deposition and systemic translocation of inhaled drug products. And especially this time, we are focusing on the dry powder inhaler. So by saying this kind of simulation, we are actually build the model based on the computational fluid dynamics and also the discrete element method. As you may see, when we say this the model is all-in-one, we started to simulate from the emitted particle size distribution coming out from dry powder inhaler, and then we want to simulate the long deposition to predict the delivery dose of those drug particles. And furthermore, we plan to simulate the systemic translocation of those therapeutic chemicals. So this is basically I will take this chance to thank all my graduate students and also all the agencies and the collaborating companies. I would like to thank Dr. Koshiyama to share their evaluate model with us, which you will see later in the slides. And I also like to thanks for the academic partnership provided by ANSYS and ESSS, which enable us to provide efficient computational work during the last couple of years. So what I will discuss today is first, I will briefly talk about why do we need this all-in-one in silico models to predict the generation, the transmission, the generation, transport, and deposition, and further translocation of those inhaled drugs. And also, because there are already many in silico models existing in our research world, so I will just talk about the two deficiencies we see from the existing models and how we want to improve from that. And then I will start to talk about the all-in-one in silico model we are developing, try to make sure that we deliver the message that the step one is what we want to predict. What we want to do is to predict the emitted particle size distribution coming out from the drug inhalers, either it's patented inhalers or it's generic inhalers or for the generic products. And the step two is definitely the prediction long deposition. Then it's about the future work. We also want to extend this long deposition and bring the simulation to what we call the health endpoints. And we want to predict the pharmacokinetic profiles too. So by saying that, when we talk about this kind of drug delivery into the long airway, usually we treat asthma. It's actually not treat asthma, but also it's to try to control the asthma or relieve the syndrome, like the COPD, those chronic lung diseases. So usually what we are targeting is to deliver those drug particles into the peripheral lung. So we try to find our way to deliver as much fraction of those particles as possible to the deeper airway. And by doing that, sometimes in silico model is quite helpful to be served as a effective way to do the parametric analysis to try to find the best combination of the coordination between patient and device or the particle characteristics to make sure that we maximize those kind of delivery. And also a couple of days ago, I got this slides from Mark. So thank you so much. So this is like the therapeutic equivalence idea, like provided by the US FDA. So when we talk about the therapeutic equivalence, and there is a part which is the bioequivalence. So by saying that, we want to show that if we have either new drug product or generic drug product, we want to make sure that they can provide a similar bioavailability to the reference at the site of action. And what are the site of the action? So when we talk about the inhaled drugs, it's always about the first step. So we want to make sure that the particle size distribution coming out from the inhalers or MDI is equivalent. So the size distribution is similar, which means that we have a good start to deliver things into our long airway. And the second step, when we talk about equivalence, is always about the long deposition. So if some drugs can deliver, let's say, a 50% into the deep long airway, and we do want some other generic products or other drugs to be delivered around the same level. But the last but not least is about the systemic translocation. So how things goes into the bloodstream and goes into our organs is also very important. So we want to show that the PK profile is more or less the same. So by saying that, our model, the step one, is always focused on the prediction of the emitted particle size distribution coming out from the inhalers. So as you may see in the slides, we may have a patented device and a generic device, and we want to compare their particle emitted patterns, especially about this particle size distribution when they exit from the mouthpiece of the drug inhaler. And by doing this kind of simulation, computational fluid dynamics is not quite sufficient because the particle suspension in the flow channel of the inhaler is quite dense. When we say dense, is about the volume fraction is very high. So the particle-particle interaction, like agglomeration, de-agglomeration, is very important. So doing this part of the simulation, we need to employ the discrete element method to explicitly model the contact force between particles. So this is very important for us to provide a accurate prediction of the particle size distribution. The second step, of course, after we got the particle size distribution, we know that what's going into our long airway from the mouth or sometimes from the nose. We want to further predict the long deposition. And we know the local deposition, we can know the regional deposition, and we want to also find equivalence of this deposition patterns, or at least regional deposition fractions should be similar between products, or let's say between different devices. So the existing in silico models, there are a lot of long models, and there's a lot of simulations about the dry powder inhaler. So there are two deficiencies we see now. So you may already see that when we simulate the dry powder inhaler, sometimes there is a capsule inside of the flow channel, and most of the simulations neglect this kind of motion, or let's say the rattling capsule motion in the channel. As we all know, that when we have the capsule moving in such a narrow channel, the movement can shift the airflow field, and which can also influence the particle trajectories and particles interactions during this transport process. So we do want to make sure that this fluid structure interaction should be captured. The second thing about the second deficiency about the existing long models is most of the long models are rigid. But we all know that when we breathe, when we do our daily breathe, it's about expansion and contraction of our long airways from time to time. And this is also very important, like a moving boundary, to help us to predict more realistic airflow field and also the particle deposition in the lung. So by trying to tackle those two things, the workflow we are trying to create is trying to do the first step as the simulation to predict the emitted particle size distribution. But in this simulation, we use CFD, DM, plus the capsule rattling motion, which is the fluid structure interaction. And the second step, we grab this emitted APSDs and predict the long deposition using an elastic whole lung model rather than a static whole lung model. And by doing this, there are a lot of validations and calibrations we need to do. And as you can see to the left of this workflow, so we may need the surface energy, emitted APSDs, experiment measurements, and also we can validate our lung deposition with in vitro test to calibrate our disease-specific airway deformation. We may also need clinical data support. So by doing this, this is more like a framework to show how we can collaborate with other experts to achieve this goal to build the all-in-one silico model, which is more accurate and more disease-specific. So by doing this, I want to show a little bit details. So when we use the hand inhaler as an example, the first step, we want to predict the emitted drug particle size distribution coming out from the inhaler. And of course, we always start from the geometry, grab the flow channel, build the mesh, and do some mesh validation and verifications by compare their airflow field and particle transport phenomena. And then we can play with the actuation flow rate, play with the particle shapes. Sometimes we can also play with the surface energies. And as you may see that you can see that how those drug particles coming out from the flow channels. And of course, those actuation flow rate should follow the draft guidance provided by US FDA or other FDAs. And it's very important to closely follow their guidance. And also, by saying about these rattling motion, this is just some preliminary work to show that what we are doing now. So we are trying to make sure that we do this kind of simulation and compare with the static simulation of the capsule motion, and to see that how this will change the particle distribution coming out from the inhaler. As a second step, we are trying to build this whole long model, which, as you may see from here, we try to simplify the long airway trees by extended like a multi-pass truncated whole long model. So in each lobe, we try to leave just one branches from the high generation to the end of the long airway, which is the alveoli. But the most important things, as you may see, we are trying to move all the long airways, so it's elastic, and so everything is moving. The good thing for us to do this is that we are able to achieve this kind of disease-specific air deformation kinematics together with the prediction of particle deposition. So how can we do that? What we want to do is try to capture what we call the anisotropic airway deformation. So usually from head to foot and back to front, there is no restrictions about our lung to expand. But from left to right, because we have the rib cage, so the deformation somehow is quite limited. So we do want to capture this anisotropic airway deformation, and you may see some equations here. And by adjust some of the coefficients in those equations, we are able to adjust the airway deformation kinematics to follow those volume changes, which are disease specific. So capturing this will give us the opportunity to use our model to predict the disease-specific patient device coordination and see how the particles can deposit more or less in the prolonged airway. Future work, of course, we want to extend the stimulation from deposition into the whole human body with the what we call the physiologically based pharmacokinetic model. So to summarize what we are doing, so we try to build somehow like improve the all-in-one modeling framework, and this can simulate inhaler, respiratory system, and the systemic regions to follow the particles from their emission to their final destination. And for this kind of new modeling framework, we are able to capture the capsule motion, and also we are able to capture the disease-specific airway deformation. So we want to make this evaluation to be disease-specific, and we can evaluate the therapeutic equivalence, and also we can help using this in silico model to improve the drug delivery, to optimize the delivery into the designated long sites. So the long-term goal, of course, I think this is shared by all the panelists and audience. So we do want to build a in silico clinical trial platform to save time, save money, and to accelerate the innovation of the pulmonary healthcare. So that's all for what I want to share. Thank you very much.
Thank you very much, Hugh. So far, as far as I can tell, I don't think there's a question in the chat, unless I'm mistaken. Maybe you can. Oh, yeah. And in the meantime, Federica is sharing the results of the poll. Maybe you can very briefly highlight, though, where your models are in terms of regulatory interactions with regulatory agencies. Would that be something that you can quickly comment on?
Mm-hmm. Sure. So all this, the design of this all-in-one modeling framework, what we do is following the FDA's recommendation, and especially about they just released a couple of the proposal announcement. So they do want to use a whole long model to predict the long position and see how this can provide more information about the somehow like the equivalence between the generic dry powder inhaler and also the patented dry powder inhaler, and to use this kind of in silico model that they can either review the application more efficiently, or if this kind of in silico model can provide it to the pharmaceutical company, they can also use this kind of model to evaluate the equivalence between their design, their design and the original design, so they can improve the design to reach that equivalence faster than just only using the in vitro test. So that's just my understanding. Yeah.
Okay, thank you very much. Then with that, I propose that we go to the second speaker, and maybe Federica, you can release the second poll in the meantime. The second speaker is from industry, from Europe. It's Alessia Beretta. She is a product manager in the cardiovascular division at Silico Trials. She is a biomedical engineer from Politecnico di Milano, and she focused on computational fluid dynamics of cardiovascular models during her studies and her PhD. Afterwards, she went to Silico Trials, and there she is responsible for cardiovascular as well as inhalation medical device projects. So really working at the interface between drugs and devices. So we're looking forward to that. Alessia, the screen is yours.
Thank you. Can you see my screen?
Yes.
Okay, great. So, hi everyone. Thank you for the invitation to be here today. So, my speech will be about computational fluid dynamics modeling and simulation for intranasal drug administrator. This was work provided together with the company Cipla. And it was about a new device for dry powder drug administration. So the background is the following. So intranasal drug administration is gaining interest from industry and also from regulatory bodies as a potential substitute for intravenous administration, thanks to its delivery to the systemic circulation that is bypassing the gastrointestinal tract. So in this project, we use the computational fluid dynamics to assist the pharmaceutical company Cipla in the design and optimization of its new device. In particular, we tested a new intranasal administration device provided by Cipla. The objective of the study were to evaluate the drug particle deposition in the nasal cavity, and in particular for this drug, the objective was to optimize the administration in the first tract of the respiratory system, so in the nasal tract, so not going into the lungs. So the objective was to maximize the deposition in the nose, and also to provide insights on how the device was functioning in terms of fluid dynamics, and finally provide recommendations on possible strategies to optimize and maximize particle deposition. So the anonymized patient DICOMs were provided by Cipla, and we performed the segmentation and the coupling between the anatomical geometry and the geometry of the device. The device was a single spray device with a complex internal geometry. So the final mesh was resulting in 5 million polyhedral elements. As boundary conditions, we defined a velocity inlet at the device inlet and the pressure outlet at the pharynx outlet. Then we had other settings regarding the coupling between particles and airflow, and also related to the device nozzle position. And all these settings were set up according also to the company protocol. And the interesting part is that we performed this simulation using ANSYS, who is one of our commercial partners, and the boundaries at the nasal walls were defined as trap boundary, which means that if the particle is hitting the wall, it means that it is deposited on the wall. So we were counting the particles trapped by the wall. And also a single drug dose spray was simulated as a particle release from an internal surface of the device. So here on the right, you can see some pictures of the mesh. We run, first of all, steady state simulations, and you can see some pictures here related to the flow streamlines within the nose. The flow here is from one nostril, so that we can see half of the nasal cavity full of streamlines, and the highest velocities were in correspondence of a specific internal component of the device, where the cross-section was the smallest. And on the right, instead, we have a clipped vision of the internal nasal cavity and the representation of the particle depositions that was maximum at the inner side of the nasal cavity. So we explored different values of inlet velocity. So we started from the injected volume that you can see in this graph. The injected volume was provided by experimental tests by the company. So you can see that there is a wide span of diameter dimensions. So from around 0.2 microns to some hundreds of microns, and this is the distribution of the formulation. We tested, first of all, the velocity, the nominal velocity that was provided also from experimental tests, and the blue line that you can see is the trapped volume resulting from simulations. So here we already see that the trapped volume is depending on the particle diameter. So above 10 microns, we see a higher volume that has been trapped. So we tested different values of inlet velocity to see the impact of the velocity of the spray to the deposition. So basically, what we could also expect is that a lower velocity corresponds to a lower trap volume. So from orange and up, we have half the nominal velocity, and going up to twice the initial velocity, we were going from 14% to 29%, with the initial velocity around 21% of trapped volume of the drug within the nose. So from these results, we observed that the particle deposition is affected by flow velocity mostly above 10 microns of diameter, while for diameters lower than 10 microns, we have almost the same behavior. So the lines are almost overlapping. So this is where we will have more effect of the spray velocity inlet. And then these results suggested that we should explore the particle deposition through transient simulation. So what happens if we have a spray velocity inlet that is not constant, but varies during time? So what we did was to simulate the inlet velocity variations as a function of time during the spray interval. So the time was divided in a spray intervals, a first period where the spray is actually performed and the velocity increases, and the second period of time when the spray is done and the person is simply inhaling, so a constant velocity of inhalation. This spray interval was simulated with different velocity functions. One was a ramp, a linear one, and then we had a power and a root function. And the result was that the root function, so the one that had highest velocities for a longer period of time, was the one with higher trap volume. This is an example for the particle diameters of 17 microns. So greater than 10 microns, where we saw that there is an interesting behavior of the particles. So we see that there is much difference between one and the other. So this spray interval, how the velocity changes during the spray interval, has actually the impact, high impact on the final trap volume. On the right, we can see an interesting graph, which represents, we can start having a look on the blue line, so related to the steady state. So this graph represents actually a characteristics of the system. So this graph is the percentage of trapped particles for each diameter dimension. So for the steady state, we saw that for diameters bigger than 10 microns, the percentage of trapped particles was increasing, reaching a threshold, a plateau around, it was about 30 microns. Consider that this is a logarithmic scale, so this is around 30 microns. So what's interesting is that the transient simulation with the ramp, the purple line, so the ramp function during the spray interval is the purple curve. And these 2 points, the green one and the red one, are these two other points. So we see that for a diameter of 17 microns, there is a very big discrepancy between all those 3 points, meaning that changing the way the spray is delivered in the nose has a high impact on how the particles are deposited or not. So the impact of the inlet velocity variations as a function of time during the spray interval was significant, but it was significant in particular for particles of a dimension between 10 and 30 microns within the C+ formulation diameter distribution that we have seen in the slide before. So what we did with this results was to provide model-driven recommendations to the company in order to increase the particle deposition, which was the objective of this project. So what we did was to recommend, for example, to increase the inlet velocity because it was correlated to higher diameter deposition, particle deposition, and it was performed, for example, by modification of the device nozzle diameter. So this results had an impact on how the device was actually manufactured, so on the geometry of the device. Second, to maximize the fraction of time at maximum inlet velocity within the spray interval, so also changing the diameter of the nozzle, of the device nozzle, and changing the functioning of how the spray is performed by the device can also change the fraction of time at maximum inlet velocity, so how the velocity is changing during this spray interval. And third, to modify the particle diameter distribution towards greater particle diameters. For example, we saw that particles with diameter smaller than 10 microns would not be very much absorbed by the nose wall. So the recommendation by the results that we obtained was to modify the diameter towards greater diameters. So to conclude, this case study demonstrated the high value of this kind of modeling to support the design of such an innovative medical device by providing insights on how particles are depositing within the nasal cavity, but also insights on the device functioning and leading to recommendations on possible strategies to maximize particle deposition. So thank you.
Thank you very much, Alicia. Are there questions. I think there is a question coming from Josh Williams, but I can't see it. Okay, there we go. There was one for the previous speaker. Maybe we can first take one for Alicia, and then ask the one to you. Alicia, you mentioned at the beginning that you do this together with Cipla. Are
Yes.
these results that you showed, are they then actually also being actively used by the company to improve their designs, or where is it maybe located in their R&D process, the use of these models?
Yeah. So as I said, the recommendations were provided to the company, and it was the phase was about the prototyping of the device. So they provided us the geometry of the first prototype to test, so that they could change the geometry of the device according to the simulation results. So it was in an early phase of the development of the device, and the recommendations that we provided were were used by Cipla to make modifications to their device.
Okay, thank you very much.
You're welcome.
Then maybe we can give now the floor for a second back to you to answer the question.
Surely. So I saw the question, and the question is about whether we consider the charge effect between the particle and Watt's interaction. So actually, although currently we do not consider that. So what we are modeling is we use the Hessian force model to consider not only the elastic force between the wall and particle, but also the adhesion. However, we do not consider the charge effect, but to adding that is not complex. So as you know, that we are using the commercial software RocketDM, so that's definitely easy to add that somehow, like the charge effect and with the electrostatic forces, and that's something we can achieve easily in the future. Yeah, thank you.
Thank you very much. And then we can go to I cannot see the question. So Alicia, there's one more question for you. How important is the particle deposition in the nasal cavity for the absorption by the body? Could we have ignored the CFD analysis and performed just PKPD from an average distribution, and would that have been very different as a result?
Okay, yeah, so in this specific case, so for the drug purposes, so the importance of the nasal absorption was very high. So that's why we didn't simulate the absorption in the lungs, for example, but we simulated the absorption in the nasal cavity, because the objective was to have an absorption in the first tract of the respiratory system, because the drug had to go very quickly to the brain. So that's why we simulated this first tract. Of course, the simulation of a PKPD analysis could be added to the simulation, and this is also what we are planning to do further to simulate then how the concentrations distribute in the system. But that's why we put so much attention on the nasal absorption.
Thank you. And the second question is, which method is used to measure initial velocity, or inlet velocity? I'm sorry.
Yeah, so the velocities were measured by the company with their bench test methods. I'm not sure I can answer specifically to which method, but these values were provided to us by Cipla.
Okay, thank you. I see there are two more questions, but we need, and for the sake of time, I propose that we go on, and maybe Some of these can actually be turned into a more general question, and we can pick them back up at the end of the workshop. So we are moving fields now from respiratory to cardiovascular, but we're staying in Italy with the next speaker, Maria Cristina Gagliardi, who is also a chemical engineer and with a PhD in chemical and materials engineering from the University of Pisa, who is an expert in cardiovascular applications and who is working for the National Research Council Institute for Nanotechnology in Italy. Maria Cristina, the floor is yours.
Yes, thank you. Okay, can you see my screen?
Yeah, sure.
Okay, so hello, I'm Maria Cristina from Italy. And in this presentation, I will talk about my contribution in the field of finite element method applied to drug-eluting stents. Drug-eluting stents are a class of medicated devices able to restore the permeability of an occluded artery in the presence of an arteriosclerotic plaque. Also, they can scaffold the arterial wall to avoid the elastic recoil of the vessel. And finally, they can provide a localized pharmacological treatment to limit the post-stent restenosis. DES can be covered with thin polymeric drug-loaded coating to control the release of the drug and maintain the drug concentration in a therapeutic range. Used polymers should be biocompatible, adherent to the metallic structure, and should have appropriate mechanical properties. In this work, we will see a numerical simulation by means of the finite element method of a stenting procedure with polymer-coated drug-eluting stent. Two cases are considered: the free expansion of the stent and the expansion in a realistic artery comprising the atherosclerotic plaque. We will calculate the mechanical stresses, and finally, we will see the drug distribution maps within biological tissues. Three commercial stents were considered here: the Palmat's Schatz, the Palmat's Genesis, and the Abbott Multilink Minivision. And one metacrylic copolymer loaded with the drug paclitaxel, that is an antimitotic drug. Stent geometries are reproduced with ProEngineer, and the finite element analysis is obtained with ANSYS. Stent deformation is simulated by modeling the inflation of a balloon angioplasty that is reported here as the blue cylinder. Here, the model is solved under the classical continuum mechanics with the hypothesis of large strain conditions. The starting diameter of the unfolded balloon was 0.6 millimeters. The balloon is assumed to be fully unfolded when its diameters reached 1 millimeter. Then the balloon is further inflated to reach the final diameter of 3.1 millimeters. That is the final diameter also of the stent, the implanted stent. The mechanical behavior of the balloon is modeled with 2 parameters Muni-Rivlin hyperelastic material law, simulating the first low stiffness phase, or the unfolding of the balloon, and after there is a higher stiffness phase. The strain energy density function, reported here with double B, is 2 parameters Muni-Rivlin, and we have I1 and I2 that are the first and the second stretching variants, respectively, while A coefficients are the Muni-Rivlin constants. The pressure inside the balloon is linearly ramped from 0 to 10 bars in 200 seconds. It is because slower stent expansion is reported to be more efficient, more performant. For the mechanical analysis, we can assume that the elastic strain is negligible in respect to the total strain. so that the total strain rate can be expressed with the additive law reported here. The stress rate can be expressed according to the classical plasticity theory, and can be described with an incremental constitutive law as reported here, sigma dot, and its associated flow rule, epsilon dot PL. Here, lambda is the hardening parameter, while the function f sigma is the plastic potential that contains the hardening function key alpha, which is here assumed to be linear and isotopic. K is the hardening modulus, and epsilon bar PL is the plastic equivalent strain. Palmat's stents are constituted of AC316 steel, while the Abbott stent is composed of a cobalt chromium alloy. So materials were both modeled with a bilinear stress-strain curve as reported here. While for the polymer, the mechanical behavior was modeled as on the basis of experimental data. Here we have the black curve, that is the true stress, true strain stress curve for the polymer, for the pure polymer, while the red curve is the same curve but for the drug-loaded material. The coronary artery is modeled as a cylinder composed of a single layer that behaves as a nonlinear, hyperelastic, and isotropic material. And the constitutive law is based on a polynomial Muni-Rivlin strain energy density function of 5 order. The atherosclerotic plaque is reconstructed on the basis of some pictures available from the web. The geometry was considered asymmetric to represent a more realistic case study. The constitutive law is based on a hyperelastic, perfectly plastic material to catch both the soft and the non-elastic deformation of the plaque. The hyperelastic part is described by this two-parameter polynomial, where J is the determinant of the elastic deformation gradient. And once the intraplaque stress reaches the values of 0.65 megapascal, the stress was imposed to remain constant for higher deformation. After the mechanical analysis, the deformed geometries were used to simulate the drug delivery. The drug delivery model is completed introducing some relating parameters, and the model takes several aspects into account, such as the diffusion, of course, across the coating, but also across the plaque and the atherosclerotic wall, the convective flow across the arterial wall due to the transmural pressure, the drug consumption related to metabolism, and finally the loss of drug in the blood flow. The diffusion was considered isotropic in the polymer and in the plaque, while it was considered orthotropic in the arterial wall. Data related to the mass transport properties of the polymer are experimentally calculated here by means of some in vitro drug delivery tests. From the releasing profile that you can see here, we can see that there was burst effect at very early times. So the function describing the diffusion coefficient is designed to take this aspect into account. In particular, it combines the early and the late diffusion coefficients that are reported here in this plot. The amount of drug lost in the blood flow was pretty difficult to be quantified, mainly because there is a lack of experimental data. So it was adopted a restriction that was reasonable because the drug was considered the drug dissolution in the blood flow, but considering only a steady state blood flow. The coefficient of dissolution was of the drug was gated by the literature, and also it was considered a complete re-endothelialization of the device after 4 months. And finally, combining all these 3 aspects, it was constructed this function for the dissolution rate in the blood that was implemented in the model. So now let's talk about the results. Here we have the von Mises equivalent stress calculated in the polymer coating. Okay, we can see the effect of drug loading in the free stent expansion, so not in the presence of the arterial wall. The drug loading decreases the stress state in the coating, and it was due to the overall decrease of mechanical properties of the polymer induced by the presence of the drug. The same effect is obtained in simulation of in contact stent expansion or in the presence of the atherosclerotic plaque and arterial wall. Here we can see the equivalent stress, once again, in the polymer coating in the plaque side or in the outer surface of the stent. Those results, also in this case, the stress state is lower in the presence of drug, and the results were generally higher than those obtained in the free expansion simulation due to the presence of the plaque that add an additional stress, external stress to the coating. However, the plastic stress in the coating is really low, and it is very localized only in small portion of the stent coating. So the total plastic strain is in all cases smaller than the strain at rupture of this material, and thus the coating integrity should be ensured. Moreover, the coating integrity was also experimentally verified by coating, by deep coating in a polymer solution, a commercial stent, and then expanding it in a laboratory procedure, not in vivo, of course, with a balloon angioplasty. The residual amount of drug in the coating is compared to experimental data. The cumulative in vitro released fraction after 24 days was around 4% of the starting loaded amount of drug, that was 5% in respect of the polymer. It indicates that the residual amount of paclitaxel in the polymer was around 4.8%, this bar here. The mean residual drug concentration calculated in the coating from numerical model was around 3.7%. That is lower than that experimentally calculated, but experimental in vitro tests are not able to catch some aspects, such as the drug advection across the arterial wall, because there is no arterial wall in vitro tests, but also the dissolution in the blood flow and the drug metabolism. Paclitaxel concentration in the atherosclerotic plaque, the picture at the top of this panel, and in the arterial wall, the picture in the bottom, is computed at a different time for each device. Here we can see the drug distribution maps for the Parmax genesis, and obtained computational results are in good accordance with experimental results reported in the literature, confirming the reliability of the proposed model. Moreover, while the short-term drug concentration is higher in the plaque than in the wall, there are some diffusion processes that move a pharmacologically significant amount of drug from the plaque toward the arterial wall, in particular in the first week after the simulated implant. At longer times, the concentration maps cover a larger area, and it means that the localized drug administration is optimized. And there are only small differences between the drug concentration and in the plaque and in the wall. Here we have the drug concentration versus time in the arterial wall. The larger drug concentration is computed in the first 2 weeks after the simulation of the implant. There are some previous experimental studies that have indicated that the vascular smooth cell proliferation is mainly occurring in the first 2 weeks after the implantation. So an optimized polymer coating should provide a drug delivery profile that is able to reproduce an arterial drug concentration similar to that computed here. Computed results are here compared to some experimental tests available in the literature in order to check if the amount of paclitaxel that is released from the coating does not reach the potentially toxic local concentration, and if it provides an effective therapeutic concentration in the artery tissues. Here we can see that the simulated releasing profile is within the effective therapeutic range for paclitaxel, at least in the first 20 days after the implantation. It could be a good result because this kind of delivery profile should be able to prevent the interstitial stenosis, but without affecting the re-endothelization of the device. So in conclusion, we have seen how to obtain a powerful finite element framework for the analysis of both mechanical and drug delivery behavior of a drug-eluting stent. The finite element analysis that was proposed here is able to check the coating integrity after the stent expansion, and also is able to give the drug distribution maps in the biological tissues. It is mainly because the in silico tests can seriously facilitate the study of the ES behavior, providing some results that cannot be otherwise gathered. Moreover, this finite element model can give good results in small times and with reduced costs, being also versatile to be customized for different stent models or other coatings, other materials, drug, and so on. So now I've finished. I would like to thank you for your kind attention.
Thank you, Maria Cristina. We have one question, and that is related to the material properties that you used, where you actually got them from or measured them from.
Material properties in terms of mechanical?
Of the biological tissues.
I don't understand if you want to know about material properties in terms of mechanical properties of mass transport.
Yeah. No, the mechanical arterial wall model, where you got the input for.
The input was from the literature, just from the literature.
Okay. Okay, thank you. With that, Federica will start the next poll, and I will introduce our senior or final speaker, our final technical speaker. We go back to the US, and Mark Harner will provide us with his point of view on drug-device interaction modeling. He's a senior principal engineer at ANSYS, also has a mechanical engineering background from Northwestern University, and he holds a number of industry leadership positions. And if you have been anywhere in this in silico modeling and credibility field the last couple of years, you will have no doubt seen him at several occasions talking about the work he has done in terms of the ASME V&V40 or in some of the other interest groups. So Mark, thank you very much for joining us, and we look very much forward to your talk.
Yep. Thanks, Liz. I appreciate it. Okay, so I'm looking to be able to share my, oh, here we go. Okay, sharing my screen.
Yes.
Perfect. Okay. Hold on, there's a big, there we go. Okay. Yes, so I was asked to talk about, well, the title of my talk is System Level Modeling of Medical Devices in Patients, but I will also spend about half my time speaking to the importance of verification and validation in computational modeling. As Liz mentioned, I'm a senior principal engineer for healthcare at ANSYS, but I listed a couple other activities that I'm involved with right now supporting the use of credible modeling and simulation. One of those is vice chair of ASME V&V40. If you're not familiar with V&V40, you'll learn about that in just a second. And the other is I'm the Global Harmonization Task Force leader for the Avicena Alliance. Okay. So let's jump in, just as an outline. I didn't put an outline together, but I'll talk a little bit about the importance of, verification and validation and establishing the credibility of computational modeling and simulation, how we can use that as evidence in our regulatory submissions, and then I'll try and bridge the gap with this drug-device interaction by I came up with a really quick example showing the application of V&V40 to one use case for an insulin pump. Okay, so this is a slide that we received that was shared with me by Tina Morrison many years ago. Some of you on the phone may have already seen it, but what she wanted to speak to here was the opportunity for computational modeling and simulation. And the observation from her was that, at FDA, they see industry and sometimes things show, evidence showing up in design history files that shows that computational modeling and simulation was used as part of the invention and prototyping phase during device development. They would see modeling and simulation used post-market, say when we're redesigning a product, going from one generation to the next. Also for root cause analysis, if there was a recall or a CAPA that needed to be addressed. But one of the things that they wanted to do here in the US was support the use of computational modeling and simulation as part of the regulatory decision-making process. But the question that comes up immediately is about verification and validation. How much credibility is required to justify the use of my computational model for regulatory decision-making? And this is important, right? So when we think about model validation, first off, we have to be able to convince ourselves as modelers that our model is credible. Then we maybe, if we're working in a device company or doing research in an academic environment, we have to be able to convince our colleagues, right? And be able to establish that that model is credible and that the results that we've achieved with that model can be trusted. But then finally, those are great for our working with ourselves or making internal decisions. But if we want to move externally to a regulator, then that ups the bar and ups the ante even a little bit more. So we really need some modeling and simulation best practices available to us to help us to, guide the development of credible models. And so then comes ASME V&V40. So what we developed, and I'll give you just a little bit of background on that over the next few slides, is what we call a risk-informed credibility assessment framework. It's quite a mouthful. We tried to come up with a catchier name. We just couldn't do it. But the idea is that, we've outlined a process for establishing the credibility requirements of a computational model based on the risk associated with making a decision based on the model. And you see that framework here. This is our figure 1 in the V&V40 standard. So what I'm going to go through over the next few slides is these first 4 elements. The rest of it's, more focused on standard V&V, as well as, credibility assessment, documentation, and evidence. But what I wanted to do is give you a quick flavor of, what are kind of the core elements that help guide the development of credible models. So within the V&V40 standard, we start off by first defining a question of interest, which describes the specific question, decision, or concern that's being addressed, based at least in part on a computational model. And then the next step is to define what we call the context of use, and that's the specific role and scope of the computational model that's used to inform that decision. And that's within the context of additional evidence. So, we may have a computational model that's helping us to answer a question, but we may also have bench testing, animal testing, clinical trial, etc. So all of that evidence that we have that feeds into answering the question of interest should be considered. Okay, the next step then is to assess the model risk. And model risk is the possibility that the model may lead to a false or incorrect conclusion about device performance, resulting in an adverse outcome. And when we talk about adverse outcomes, we could be, obviously talking about the patient, but there could also be things like a clinical outcome that's adverse, such as a clinician has to go in and do an intervention, re-intervene, maybe something breaks free from a delivery device, they have to go retrieve that, etc. Or you may also have adverse outcomes on the business. So it could adversely somehow roll up to impact the reputation of the business. So the 2 elements of our model risk are model influence and decision consequence. So model influence is a contribution of the computational model to the decision relative to other available evidence. So here I have a couple pie charts on the x-axis, and they show the relative contribution of bench testing, animal testing, clinical trials, and in silico. And as you go from left to right, the blue piece of the pie gets bigger, right? So we're relying more and more on the computational model to make our decision. If we talk about decision consequence, now decision consequence is the significance of an adverse outcome resulting from an incorrect decision. So if we're making something like a reflex tester, if there's an adverse outcome, it doesn't quite work correctly, well, it's going to impact the reputation a little bit, well, probably a lot, because it's a really simple device. But at the same time, we're not going to harm the patient or the clinician. So we would call that decision consequence typically pretty low. Whereas if we have a problem with our blood pump that's supporting a patient while getting bypass surgery, or maybe it's being used as a bridge to transplant, that decision consequence is much higher, right? The impact on the patient is much higher. So what we have to do then is bring those 2 elements together to come up with an overall model risk. And you can see here that even though we're relying very heavily on modeling and simulation to develop a reflex tester, the overall model risk is lower than for the blood pump, where even though we're relying very little on computational modeling relative to the other available evidence, the risk associated with that computational model is still higher. And because it's higher, that means we have to spend more time validating our computational model to ensure the credibility of that model. And so the last step then that I'll go through in the V&V40 standard is to talk about credibility goals. So model credibility refers to the trust and the predictive capability of the computational model for the context of use. And we establish that trust through the collection of evidence, right, the V&V evidence. In addition to that, though, we also have to demonstrate the applicability of that evidence to the question of interest. So it could be great if you make this really nice validated model, but if that evidence you generate isn't relevant for the question we're trying to answer, it's like, okay, so what, right? So then the idea is that based on the risk associated with the model, a low-risk model will require lower credibility than a high-risk model, okay? And so what we do in this case is we subdivided the verification and validation process into its constituent elements, which we call credibility factors. And so for the credibility factors, we have things like software quality assurance, numerical code verification, discretization error, etc. Okay? So the idea is you go through these one by one individually, evaluate the evidence that you require to establish a sufficiently credible model. And this then gives us, a framework for having this discussion about how credible is our model, and have we developed a sufficiently credible model after we're done going through the V&V process. So what that leads us to then is a regulatory framework for computational modeling here in the United States. And so what we have is FDA, luckily, focusing on this topic of developing computational modeling technologies to support regulatory decision-making. This is one of their top 10 regulatory science priorities within the Center for Devices and Radiological Health. And so they're one of our key partners, right? if they hadn't established this as a regulatory science priority back in 2011, we'd be having a very different conversation today. So they're our partner. Now, what are the steps in that framework? The first step is to go through the ASME V&V40 standard and determine where the credibility requirements of our computational model. Once we've done that, we can then go to other ASME and other standards which tell us how to validate computational models. They give us that understanding of VBUQ science. How do we do it? And those are documents such as ASME V&V10 and ASME V&V20. And then finally, once we've developed our credible model, we need to summarize that model in a modeling and simulation report that we share with the regulator. And so back in 2016, FDA published a guidance document that outlines, what are the expected contents of a modeling and simulation report. And the idea here is that if they see consistency in the information that's being transmitted to the FDA that summarizes the model, then hopefully they're able to provide more consistency and predictability in the review of that modeling and simulation evidence. Okay? And so one of the things we're working on now within the, and why I mentioned it before, one of the things we're working on now is under the auspices of the Avicenna Alliance is scaling out this framework globally. So that we can use this modeling and simulation evidence in a more predictable and consistent way in other geographies. Then I have a few slides on an example. So in this case, we'll look at one of the failure modes of insulin pumps. And so in this case, what we're worried about is whether or not a patient is going to receive the expected amount of insulin therapy while they're, wearing their wearable device. And a lot of these devices now, you kind of set it and forget it, right? They're so automated that you're not necessarily always tracking, how much is being injected, how much is going in. Now, I personally don't have diabetes, so hopefully I'm not, not giving enough credit to how much work it really is to wear one of these pumps. But I'll just say, they are highly automated, and that brings a lot more freedom to patients, a lot more confidence in the use of these devices. One of the failure modes that we do encounter with these devices is occlusion. So you can have buildup of material at the tip of the cannula that is inserted into the patient. And we can also have crimping of a catheter. If you bend over, potentially these catheters can be crimped. And that can interrupt the delivery of drug to the patient. And that can lead to adverse outcomes such as hyperglycemia, where you're building up too much sugar in the blood because we're not getting our insulin therapy. And there's also this chance of hypoglycemia, which is really interesting. So in that case, if we do have a crimp or an occlusion and it's suddenly released, there's a pressure buildup behind that, and suddenly you can have a huge bolus of insulin that's given to the patient, and now their blood sugar can be too low. And both of these are, very, poor outcomes in terms of delivering the therapy to the patient. So let's look at V&V40. Can we develop a model that helps us to understand whether or not there's an occlusion present in the system that's precluding the patient from getting the insulin therapy? So in this case, just as an example of a question of interest, within the V&V40 framework, we could say, will the infusion pump warn the patient of a flow path occlusion that is interfering with insulin delivery? Okay, very simple. Now, what's the context of use? And here are a few crib notes here. If you kind of think, well, this definition of a context of use is kind of vague, what goes into it? You can think of this as kind of the various elements that you need to include in a good context of use statement. So in this case, for our insulin therapy, we'll say that one-way fluid structure interaction modeling will be used to predict the relationship between fluid pressure, flow rate, and catheter bend angle. And the patient also has a continuous glucose meter available as part of their insulin therapy. So, we're going to use a computational model to help us to predict occlusion, but the patient does have other evidence available to them, right? They are able to track their own blood glucose levels over time. And I apologize if my voice is a little shaky. I'm in a coffee shop giving this talk, and it is freezing cold in here. I apologize. So now let's do the risk analysis. So for model influence, in this case, we're going to say the model influence is medium, since there are other sources of information available for predicting occlusion. In this case, we have a continuous glucose meter, as I mentioned before, that's monitoring our blood glucose level, and that's indicative of whether or not we're receiving the therapy. For the decision consequence, we also say that's medium, since patients can quickly drift away from the target blood glucose concentration, but can administer a correction bolus to return to that normal glycemic state. So if we sit in medium in the model influence and medium in decision consequence, then our overall model risk for this scenario is medium. So what does that model look like? Let's now take it away from V&V40 and say when we're talking about the drug device interaction workshop and what we're talking about here, how can we use drug device interaction modeling to help us support development of credible models and simulations? So in this case, we're going to do a pressure flow analysis, as I mentioned, in the context of use. So we could develop a reduced order model in this case that represents the relationship between catheter bend angle, flow rate, and pressure rise. And we're going to develop that using a set of one-way fluid structure interaction simulations that I mentioned before. And so in this case, what we have is a straight catheter that we bend using finite element analysis. And of course, we use ANSYS for this. We take that catheter, that bent catheter configuration, we export that into Fluent, and then run a series of pressure flow analyses that establish for us a relationship between flow rate and pressure as a function of the bend angle. Now you see three different scenarios here, but what we can do is, create a family of one-way FSI simulations where we look at a range of flow rates and a range of bend angles, and we put together a reduced order model representation that tells us that relationship between pressure, angle, and flow rate. And then what we can do is incorporate that into a systems model of the drug delivery subsystem of the insulin pump. So in this case, we have a multi-domain model that represents the embedded software domain, the electrical domain, and the fluids and mechanical domain. You can see the ROM here from Fluent that's telling us that relationship between bend angle, flow rate, and pressure. And we can use this kind of system level modeling and simulation to understand the interaction between the fluid mechanics, the electrical system, the control system, and the embedded software. Because what we want to be able to do is virtually run, essentially in silico trials that tell us, as a function of the different flow conditions downstream and different levels of occlusion, which we can control with this ROM, as that rolls back up into the cannula, the catheter, the electrical system, the control system, and eventually the embedded software, we want to know the embedded software will warn us that there is an occlusion of the catheter when that occurs. And what is the delay? How will that embedded software perform? And that's what's really exciting now, is that through model-based systems engineering, we can start to go into much more detailed levels of simulation that help us understand from a multi-physics, multi-domain, and multi-scale level, how will our system ultimately perform? And then to take that to an even higher level, and again, I'm going kind of quickly and really taking this a high-level view. There's a lot, we've done all this work. All this has been done. I just don't have time to go through it. But we can also incorporate pharmacokinetic modeling along the lines of what you talked about earlier on, and even the previous talk of the drug-eluting stent. That's PK modeling as well when we're looking at deposition within the arterial vessel. We can do this pharmacokinetic modeling, connect that to the system-level model to help us understand, what will the patient experience be. If there is an occlusion and interruption of the therapy, and then we release that and we're able to deliver drug to the patient again, they will go potentially out of that normal glycemic state. How long will it take for the patient to come back to normal glycemic state again? We can couple the system level model to the pharmacokinetic model to really understand that the interaction between the device and the drug and the patient. So with that, I'll give you three conclusions. The US FDA has established a framework for using computational modeling and simulation as part of regulatory decision-making, and that's supporting more predictability and consistency in the use of modeling and simulation within regulatory submissions. The ASME V&V40 standard guides users in the development of sufficiently credible models. And one thing to keep in mind is we can always validate a model better. A model can always be better. We can always reduce the error bar and the difference between a model and our comparator, our experiment. The question is, within industry and academia, when we have deadlines that we need to meet to deliver our research, when do we stop? When is our model good enough? And good enough maybe sounds negative, but, and that's why we use this term, I think, sufficiently credible. But it's important to be able to know when we can deliver those results. And then, again, coming back to that last part of my talk, multi-physics, multi-scale, and multi-domain modeling and simulation enables a more holistic understanding of not only device performance, but also patient response and patient safety. So with that, I'll thank you for your attention.
Thank you, Mark. Thank you very much. There is a question from the audience. If we are using FEA and bench tests to predict fatigue safety of stents, can we rank the risk as medium to high?
Good. Okay. Okay. These always lead to longer discussions, right? Mm-hmm. So the question is, in that case, when we finally make our final decision about fatigue safety, one common way that modeling and simulation is used is to identify worst case scenarios. So what you might say is, in one scenario is that I'm going to look at my shortest stent, my longest stent, I'm going to look at the smallest crimp diameter and the largest crimp diameter, and do that four corners test within the FEA tool to decide what's my worst case scenario. And then based on the FEA tool, I will eventually go and do the experiment. So instead of having to do 4 experiments or multiple experiments, even within that space of small diameter, large diameter, small expansion, large expansion, I'm going to reduce that experimentation through modeling and simulation. In that case, I would argue that you're down into the low to medium phase because eventually you are doing a test. You are finally using the in vitro test to validate the fatigue safety of the device. Okay. Now, if you take a different approach to it and you say, you know what, I'm only going to use modeling and simulation to make that prediction, then you're moving into the medium to high phase because you don't have that final bench test that's going to tell you, yes, I'm done, good to go. So it depends, right? It depends on, you know, how you approach that problem and how you break it down. But hopefully that's helpful. Yep.
Thank you very much. And then maybe one question in preparation of the next speakers. When you would present a model like this to FDA, which has a component of drug, or the last one you talked about, when there is PK/PD, when it has a component of drug and it has a component of device, where do you go to? Or is there an intergroup or an I don't know how it's organized.
Yeah. At FDA, I know they look at the device, you know, they look at the combination product first and they decide what's the primary driver, what's the dominant, you know, element of this. Is it the drug side or is it the device side? So based on that conversation, they decide who's running the show. But that said, there'll always be someone from the drug side and device side who's, you know, involved, right? You know, I will say that right now the framework that I showed you is more fine-tuned on the device side. But it's really interesting, you know, we held a workshop a couple of years ago where we had folks both from the device side of FDA as well as the drug side. And they've actually been supporting the use of modeling and simulation for much longer within the FDA. What I would say, though, is that just that this formalized framework was not necessarily there and it wasn't a topic of discussion. I think for them, it was more natural, right? Like pharmacokinetic modeling and simulation has been used for years. And one really interesting comment I got was that FDA even recreate some pharmacokinetic models themselves in-house. They say, well, you know, the scientist was telling me, well, sometimes we're not sure. We want to double check, so we'll just recreate it and run it ourselves in-house. And that blew me away. Like, I couldn't imagine FDA saying, oh, I'm not sure about your FEA model. I'm just going to model your stent myself and let you know how we feel about it, right? It's just very different. So yeah, I'm not sure I gave the complete answer, but some insight hopefully is helpful.
Very helpful. Thank you very much, Mark.
Yep.
And then
Thank you.
It's time for the next poll and the final speakers of today, which are two ladies from the regulatory agencies. We have Ines Kotheim-Russen here from the Norwegian Medicines Agency, and Flora Muswamba Chinanu, who's from the Belgian Regulatory Agency. And they're both chair, vice chair member of the Modeling and Simulation Working Party of EMA. So they are excellently placed to provide the point of view, the currently regulatory status, or the current regulatory framework for modeling and simulation at the various regulatory agencies, and to give us their take on the issue of drug-device interaction. Ladies, the floor is yours.
So I'm just going to check that you see the presentation, because I'm...
Not yet.
Because I didn't receive the request to share. Can you see it now?
Oh, I think, Federica, I'm seeing the show my screen. Yeah, now it's there.
So perfect. Now you see it. Thank you.
Yeah.
Okay, thank you. And thanks so much for the invitation to present here. I don't know how to... Can I duck this here? Sorry about the technical. No. So I lost the screen. Did you as well, the slides?
No, we see everything, and we see your mouse moving as well.
Yeah, but I can't see my slides, so, and that would be perfect. So sorry, and thank you again for the invitation to present the regulatory perspectives here. We're very happy to be here, both Flora and I. And we're really on the drug side, so we will continue some of the talk on the credibility framework, as Mark already started, but from a little different perspective. So just first, the official disclaimer that the views we present today cannot be quoted as being made officially by any of the agencies that we work for. And the outline of our talk is sort of in two parts, and the first part is very general on the way that we already use models to inform drug development today, and sort of our perspective and the way that we try to look at models as a method to answer questions, and sort of then onwards to the credibility framework, as Mark already has presented to you and many of you already are familiar with. And then Flora will take over the presentation and give 2 examples on drug-device interaction modeling that we have looked at from the regulatory perspective from the drug side. And as already said, sort of modeling has been sort of a key part of informing drug development for many years, probably the last 30 years and before, within the industry and the sponsors, but also on the regulatory side, sort of these PK/PD models and the mechanistic PK models are really sort of key part of development. And almost for every marketing approval application we receive, there will be some models that are actually informing the regulatory decision making already. And this term, model-informed drug development, the MIDD, is really sort of a key term that we use all the time, and sort of really a bit of a paradigm of how we look at models on the drug side. And the very typical applications for models on the drug domain is to inform the design of the trials, the study designs, and using clinical trial simulations and all these tools we have, but also very often to answer questions on the dosing regimen of the drug, and that could be in the sort of general target population, but also in smaller subpopulations that in some way deviate from the mean of the general population. And we see that sort of really increasingly the use of modeling and simulation as a tool to gather the knowledge we already have is really sort of making an impact on how we go about populations where there really is sort of feasibility issues. You have, for instance, in the orphan indication or in the pediatrics, you really have too few patients to do the randomized controlled study that is sort of the gold standard in drug development. And then what do you do? And then you have to really look at the knowledge that you already have, and modeling and simulation, of course, is a really powerful tool to get that together. So we really see sort of an increase in modeling being used to answer some sort of bigger questions on the development there. And what is also a very sort of typical feature of modeling in the drug domain is that you don't have one model answering one question, but you have sort of the same model being used several times during the individual development. And we also see that sort of these iterations of learn and confirm, they are really sort of helpful in building the credibility evidence and finally being able to use the model to inform some of the regulatory decisions. Still, we see that there are some barriers to the larger acceptability of models to inform decision-making, and particularly then for the high-impact applications. And there are, of course, many sort of aspects and reasons for that. And of course, sort of the data that we have and the knowledge that we have is a big part of that. But also we see that sort of the lack of standards and the best practice for how to do the modeling exercise is an important part of it. And I think in the drug domain, we have really good guidance on the empirical population PK models, PD models, and so on. But on the mechanistic sides and the more advanced disease models, I think there's really a gap on the framework supporting the modeling exercise to make sure that everything is done in a manner that enables the model to actually inform in the end the regulatory decision making. And we think also sort of the communication is an issue here. And sometimes it comes down to the terminology, and we see there are some differences in the way the terms are used from for instance, from devices to drugs, and for the empirical models to the mechanistic models. And this can sometimes lead to some confusion if you don't know about this. And also the different perspectives that people are coming to the table with can make things a bit difficult to really understand what the model is about and how you can trust it. And of course, maybe the biggest challenge there is sort of the dialogue between the modelers and the non-modelers. And talking sometimes with the clinicians is difficult to sort of get the main message across about the boundaries of what you can use the model for. And of course, then the level of detail to provide on the model. We see sort of frequently challenges there, that sometimes there's too much technical details and plots on how the model performs, and then sort of the clinical expert is a bit blown away and don't know what to do about it. And other times we really get too little information in the reporting so that we are not able to really assess what has been done and whether or not we can trust the model for the use that is being proposed. And kind of summing it a bit up and going back to this term, the model-informed drug development, because we this is really the way we are thinking at the drug side of it, and kind of sum that up as an approach of using the model as a backbone of knowledge, that you really use your in silico model to consolidate the knowledge you have on the drug, the product, the device, the disease, the patient, and use that knowledge and that model to inform the next step. And then you kind of iterate, and the next sort of study you perform, you get more knowledge and you can sort of update your model and see how you go about it. And I think already today we have very open dialogue between the sponsors and the regulators on trying to find potential applications for these models. But then we think that sort of when you have found that specific application that you want to use the model for, then you really need sort of a shift in your focus, and you have to be able to very clearly understand how and whether you can trust that model. And this is where it becomes extremely helpful to look at the model as a method, and as a method to answer a specific question. And this is where we also see sort of from the drug side that this credibility framework, the risk-informed credibility assessment is very helpful. It's a very helpful tool. And I won't go into details as it, and Mark already have explained it. But I think sort of, as he said, this was originally proposed for the medical device models. But we're now also testing it sort of in the drug space and trying to see whether we find it useful when we assess the models that are submitted for medicinal products. And today we sort of also see that sort of the models presented here and on trying to sort of merge the models and move from the device to the drug, it's equally applicable. And I think that's sort of kind of the strength of the framework, that is really general. Maybe you could say that it's a sort of general umbrella that can be used irrespective of the type of model and the type of application, because the framework as such is really outlining the process of model assessment. And this is what makes it very useful and can potentially help us harmonize a bit our approaches to model assessment. And of course, the starting point of the framework is the questions of interest. And sort of in our view, we see that this step 2, trying to establish the risk-informed credibility, it's really about being very clear and transparent about the interface between what we want to know and the method that is proposed used to provide the answer. And sort of looking at the framework from this angle, you can actually say that this interface part of it is very useful irrespective of the model or the method that you're using. Even if you propose to investigate and answer the question by conducting a randomized controlled trial, it's very useful to fill out sort of these points, because you understand more about that method and how it is able to answer the question. And we just try to sort of draw this a bit clearer here, that and sort of in our mind we kind of call this step 2 the credibility matrix, when you sort of get together all the information that actually bridges your model and the specific application, because many of the models can be used for many applications, but when you're answering a question, you're really sort of normally picking one specific application. And then sort of this step 2, or the credibility matrix, is really merging the question directly to the decision making that is sort of has to be made at the regulatory side. And you very clearly understand what is the input of the model to that decision making versus other data sources or other methods that is also available to answer the question. And sort of these things all together will form the decision making. And also, we think that when you have the very clear view on this, it's extremely helpful to facilitate the dialogue with other domain experts. So it makes it much easier to communicate from the modelers to the non-modelers, and also sort of from the modelers in the industry side or the sponsor side to the regulators. So we find this very useful. And as Mark already described it, it makes it much more clear to understand what rigor you need to present for the credibility evidence on the verification and the validation. So this is kind of our take on this step 2, the credibility matrix. We adopted it a bit to the drug space and try to implement it in the way that we already are assessing the population PK/PD models and all the models that we are normally seeing. And we find it's, again, sort of a small tool to try to make a very visual and very sort of clear overview of the modeling exercise. And we kind of did the same with the verification and validation activities to try to have some tables to really sort of quickly get an overview of what have been done, and then sort of to go into the full reports and dig into it, but really to sort of try to sort of have communication tools on what the status is at the of the model and the modeling exercise at the time it is presented to the regulators. So with that, I'll give the word over to Flora. You would just let me know when I should change the slide, Flora.
We can't hear you yet, Flora. No, we can't hear you.
Is it better now? Can you hear me?
Now we can hear you, yeah.
Yeah, I think you know this mute button that we always forget. So thank you, Ina, for a very good presentation. I think the flow between the three talks is very nice here. I would go concretely to some example that we have seen in the regulatory applications, and just to show how complex can be the situation in the EU regulatory framework. The committees that received modeling-related questions are very variable. The regulatory procedure where modeling-related questions are included are also very variable. And then we have one modeling information working party, which is there sort of to give support, but the people we're talking to have different background, and most of people in these different committees are non-modelers. Next slide, please. So I'm going to run through two different examples. Ina, you can move to the next. Two different examples that are very simple examples, and they were received as part of the Scientific Advice Working Party. And the reason why I have selected actually this example is because they are very similar in terms of the question that the company were trying to address. The application is almost the same, but you will see that the proposal that we received were very different, and having the credibility matrix in mind, having these risk-informed credibility assessment in mind, it also helps to build some consistency, and you will see how we have come as regulator with something very consistent as an output based on the similarity in the question credibility-wise. Move to the next slide, please, Ina. Yes, thank you very much, Liz. Yes, can you move to the next slide, please? So this is the first example, which is a drug-device combination. It is actually an auto-injector that the company was proposing to develop to facilitate the administration of the products by healthcare professional. The product by itself, the medicinal product, was a monoclonal antibody that was dedicated, you know, to handle the inflammatory part of COPD patients. And the applicant has conducted actually the phase 3 drug development program using a prefilled syringe formulation. And after that, for the reason that I've just mentioned, they were trying now to move from the syringe to the auto-injector. And they came now to EMA to ask for scientific advice on different questions, but the questions that are maybe relevant for this workshop were the one related to how to establish PK comparability between the prefilled syringe, which was used till the end of the clinical development, and the auto-injector that they are planning to use after market. Next slide, please. So this was exactly the question that we did receive from the company. So it was very clear that this company was planning to conduct a bioequivalence PK comparability study, which is sort of the gold standard way of proving a PK comparability between two different formulations when you are, for instance, coming for generic or this kind of thing. So normally we are for a PK study. Next slide, please. So the company came with that proposal that they wanted to conduct this PK study, that it was a PK comparability study, and it was a fully powered PK study actually, and because this monoclonal antibody has a very long half-life, it would have been a very, very heavy study. Next slide, please. Thinking about it, we did realize that actually there were not that big difference, so to speak, between the PK to be expected from the auto-injector, which would be used aftermarket, and the prefilled syringe, which was used during the drug development. To our view, there was some room for doing some modeling, like collecting some sparse sample, developing a pop PK model or a PBPK model to be verified using some sparse sample in the two formulations that could help to do some sort of model-informed bioequivalence testing. Next slide, please. So what we had in mind actually was much more lighter than what the company was proposing. They were proposing a very big trial, and this is the answer that we provided to the company, telling them that no, normally for answering this kind of question, we think there is an alternative method, which would be a model-based approach, perform a small study with sparse sampling, develop a PK model, and then you can use that PK model to implement a sort of simulation-based bioequivalence testing, like virtual bioequivalence studies, establish the acceptability criteria based on your model parameter, estimate, and precision. And we think that this is a way out. This is the official answer that you see copy-paste on the slide. Next slide, please. So just to show you how we build up that rationale, we thought that for the question that was on the table, which is whether or not we could compare the absorption profile between the two formulations, yes, bioequivalence study is the gold standard now, which is the most used one. Of course, no regulator, at least no one at EMA would say no. If this company had come directly with the result of their bioequivalence study, we would accept that. But we think that in terms of strength of evidence, the model-based approach could be acceptable as well. And then we try to push the company to do the lighter one for the patient, and because we believe that it is equally informative. Next slide, please. And then we had another company. They were also developing exactly the same drug device products for another indication. Next slide. But the rationale was exactly the same. They also developed their drug during the drug development using a prefilled syringe. And then at the end of the phase 3, when the phase 3 already completed, they decided to go for an auto-injector after marketing authorization. And then they think that for patient convenience, for healthcare provider, this would be much easier. Next slide. And they also, at approximately the same moment, decided to come and ask for scientific advice. But they had another idea, which is completely the opposite of the previous one. They were asking if possible they could put the auto-injector on the market without doing any trial at all. Then they had a very long argumentation trying to convince us that it is possible, given what is already known about the delivery system, given what is already known about the PK injection time and viability, so on and so forth. Normally, they don't need to do anything at all. Next slide, please. They did develop a POP-PK and even a PK-PD model, only using the prefilled syringe data. And they were saying that given that what they observed in that POP-PK model, they did some sort of sensitivity analysis that I would definitely not go in detail here, but it was really wanting to take, the shortcut. And this was actually their proposal. This question reminded us exactly the same that we received from the other company that wanted to perform that fully powered study. To us, it was exactly the same question. It was exactly the same application. So we provided exactly the same answer. Next slide, please. We said, we believe that there is some potential for modeling and simulation here. We would not request a fully powered study here to bridge for bioequivalence, but we believe that a POP-PK model or a PBPK model that would be validated by some data from both formulations. This data could be sparse, could bring enough confidence for the application you're claiming. And if you have that, then this could be acceptable as a waiver to have the fully powered bioequivalence study. Next slide, please. So in terms of the credibility matrix table, it was exactly the same. The impact to us was high because we're talking about waiving a very big study using model-based approach. But we believe that in order to believe that this model is credible, it will need some external validation data. And these external validation data would come for some sparse data. What we are proposing, the company is, you already have some data for the prefilled syringe. What needs to be collected now is some data also from the auto-injector to show that your model you're claiming is able to predict data from both formulations. Having that model, you can go virtual for the bioequivalence study and establish that the two formulations are bringing exactly the same PK to the patient. Next slide, please. So we are trying already to use this credibility approach to apply that to drug device, to apply that to drug development also. But as Ina just said, this is exactly the slide where Ina ended, saying that it is all in the process of regulatory decision making. The table as such is not solving all the problem, is not the whole story. After we have at the table to establish consistency, to really locate where are we in terms of the question that the model is addressing, we have a kind of assessment step in there that lead to a much more objective and consistent regulatory outcome. Thank you for your attention.
Thank you very much, Flora. I'll stop sharing my screen. And so we, given that we have run over a few times or shortly run over in a few times, I think we can go to we still have some time to do some general questions and comments related to the actual topic that we're here for today. How do we start now in this domain of advancing the regulatory science related to drug-device interaction? And if I maybe may make a first observation, it is quite clear what we've seen today, what the background of the people was. I mean, the people from a more engineering background have talked a lot about the mechanics and that kind of models on the device side, whereas then the regulators come with a more pharma background have talked about the PK side. So indeed, also as Mark indicated, when you look at a device, there is a leading drug or device site, and that will then deal with most of the regulatory aspects. So how do we make sure that in the future, maybe the vocabulary doesn't become the same, but that when we talk about drug-device interaction, that there is a bit more balance? I wouldn't say, of course, every application has a weight on one of the two sides probably, but then I think people through the methods that they're using and the background that they have tend to overemphasize one of the two aspects. Is there a need to balance this out better, or is it really not much of an issue? Maybe Flora, Ina, you can have some thoughts on this.
Yes, my answer will be a little bit biased, you know. But to me, it is not that big of an issue, actually. I mean, because it depends on what if the end user like story, what is the end application, and if we want to have the device right and the drug right, and if we all agree on the fact that this framework is actually, as Ina was saying, we are now stronger and stronger convinced that actually the framework can even go beyond modeling. So it can be applicable to drug, can be applicable to devices, it can be applicable to drug devices products. So it can even facilitate the discussion between the drug side and the device side. So to me, it's not that big issue if we really know exactly what is the question of interest that the model is kind of addressing. Thank you.
Okay, thank you. And maybe I can follow up with a question that we got from the survey, and that is a bit related to the spirit of the triple helix discussion. Is there any plan from the regulator side to start with a technical group that will involve academia, industry, and regulators to work specifically on this topic of the drug-device interaction? Is there an interest? Of course, we're here to start a process, but how should we continue this? What are thoughts on the way forward?
Maybe we can start from where we started from, actually, where the V&V is starting. That, I mean, it's all about, first of all, identifying the questions that are common to both sides, that have high impact, high interest from the drug side and from the device side, and then try to bring our force together on that. I mean, on the principle, at least, as Ina said, I could only speak for myself and say I welcome that. I really see that this is an example where we have learned so much from each other, you know, and someone from FDA was explaining us as the interaction took place within FDA, from the drug department to the device department, exchanging to end up with the V&V that was positioned for devices and now coming back to the drug space. And you see that there are some efforts from EMA, from FDA to really try to convince people to use that. So if we can go back together, to me, on the principle, I agree, but to have it run, we need to identify some commonality, some common struggle, some common question, so that we have something concrete to work on. Maybe I should leave it over to Ina.
Thank you, Flora. No, I fully agree with you, and I think it differs a bit across Europe, sort of if the devices is in a separate sort of agency versus the drug agency. But I think it's sort of the time is ripe to try to sort of establish the connections and try to look at the questions and sort of the applications that are there, and have sort of increased the flow of information, because I'm very certain that there are sort of big advances to be made to harmonize a bit and sort of to have better dialogue. So I think there are certainly some opportunities, and I think things are moving, so it's a good time to find sort of all the approach that there is to have this dialogue across sort of the academia, building sort of the models that potentially can be used by industry, and will sort of at some end up at the regulatory table. And I don't think we have so many examples now, but we see them coming. And this is also why it's very interesting for us to be here today to see what's moving, and also sort of to follow that and see sort of how we can increase the dialogue along the way.
Yeah, and to that, in connection to
Liz,
that, yes.
This is Mark. Could I throw in an additional comment there? You know, I would suggest that maybe a way to bring together these different stakeholders would be through ASME. The V&V40 subcommittee is seeing more interest from pharma in ASME V&V40. So we're seeing a real increase in pharmaceutical companies now who are saying, hey, we would like to learn more. We see the importance of V&V. We've heard of V&V40. Let's learn more. I've seen a couple papers appear in the literature where folks have, taken a pharma manufacturing process, if I remember correctly, and tried to apply the V&V40 framework to it. Based on what I've been seeing, I suggested that at our upcoming standards committee meeting, which I'm also a member of for ASME, that we at least discuss forming either a subgroup of V&V40 that looks at the pharmaceutical industry in particular. Also, something I wasn't able to cover in my talk is that the ASME V&V Standards Committee also covers other industries. So we have standards committees focused on nuclear, automotive, advanced manufacturing, AI and machine learning. Another opportunity where we could bring all these stakeholders together very easily because they're set up for it is a V&V for pharma subcommittee. So just throw that out there now, and if anyone would like to chat more or would like to help me formulate the development of that group, put together, what would be the scope, initial pitch, to ASME in terms of what we might focus on, I think that'd be a great way to just kick off this kind of effort. Yeah.
Thanks, Mark. And maybe if I can make a remark, if I remember correctly, in the ASME V&V40 committee, there were very few academics present. It was mostly industry and the regulator at that time. If I look at the people present here today, it's really 30-30-30. It's 30% academia, 30% regulatory, and the rest are industry and others. So I think the awareness in all stakeholder groups is increasing. And also the presence here is divided between drug and device mostly, and people are working on modeling across the board, across the pipeline. So I think the community is there, and I think both the VPHi, but also the Alliance, and everybody represented here has indicated that they're really willing to collaborate on this. So this will definitely be continued.
Can I give another data point, Liz? Is that, sorry to interrupt, but I know we're running out of time, but the other thing that's interesting here, because you bring up the 30-30-30, right? So yeah, we didn't preclude the inclusion of academics. They just didn't, I guess, get excited to join V&V40. There's no reason why they couldn't. The other thing that wasn't part of V&V40, and I would argue is the opposite of what we see here today, is it's primarily North American people, scientists who participated in V&V40. So it's flipped here. This is primarily European audience, and I saw 8% US. So getting more of the European community involved as well would also, I think, bring more, awareness of V&V, and we're trying, through VPH and Avicena to disseminate V&V40, in Europe and the rest of the world. But yeah, getting direct involvement. Who knows? Maybe the V&V for Pharma Standards Committee is more based in Europe. I think that'd be an excellent step forward.
Okay, thank you very much. I think indeed, being conscious of the time, with no more questions coming through the general chat, I thank everybody, all the panelists, for speaking, for giving an excellent overview of the current status, for all the participants being here. We will bother you in the future with additional questions, and we look very much forward to continuing this conversation. Thank you very much, everybody.
Thank you, Liz. Thanks for organizing. It was very nice. Thank you.
Thank you.
Thank you.
Thanks so much.